Okay, so welcome to this next video in which we're discussing angiogenesis in chronic inflammation. Okay, so we're just having a revision of both the innate immune response and the adaptive immune response. We're still on the innate response at the moment, so we've discussed that in the innate immune response, which is also known as the acute inflammatory response, what happens is you get vasodilatation of the arterioles, leading to the affected area, which leads to increased blood supply there. And why do we want increased blood supply? Well, it's so that we can take proteins and cells from the blood and move them into the interstitial fluid to fight this pathogen. Okay, so we increase vascular permeability of the capillaries and the post-capillary venules by creating gaps in between neighbouring endothelial cells in those blood vessels. Okay, that allows fluid from within the blood to move out of the bloodstream into the interstitial fluid, and there what it will do is it will fight the pathogen, because within this fluid you are going to bring certain proteins that are within the bloodstream. So you're going to bring complement proteins, and you're also going to bring coagulation factors. Complement proteins will set up the complement cascades on the surface of the microbes, which will lead to the destruction of the microbes. And in addition, coagulation factors will set off the coagulation cascades as soon as they're moved into the interstitial fluid, and that will trigger the production of a fibrin meshwork, which will encapsulate the microbes and trap them and stop them from being able to spread, basically. Then you're also going to get leukocyte recruitment, and this will occur in the capillaries and the post-capillary venules. So the endothelial cells will become sticky for leukocytes, basically, and the first leukocyte that you're going to start recruiting is the neutrophil. So neutrophils will come and stick onto endothelial cells in the capillaries and the post-capillary venules, and then what will happen is a process known as diapedesis, which I'll just summarize. Okay, so if we have two endothelial cells here, okay, then we've already discussed that as part of the acute inflammatory response, what's going to happen is these two endothelial cells are going to make a gap in between them. But I said that this gap was not big enough for cells to actually move through, and it's not, at least not without help. So the cells have to firstly adhere to the endothelial cells, okay, and they then have to be guided through this hole, and they have to sort of slither, basically, like a snake through the hole. Okay, so they have to sort of deform themselves in order to actually get through this hole. So it's an uncomfortable squash, basically. Okay, and this process of the neutrophils, or indeed any old leukocyte, because all leukocytes are going to do this, it's not just neutrophils. This process is known as diapedesis. Okay, so diapedesis is this word to describe this sort of slithering through the holes. Okay, and the movement of the leukocyte from the bloodstream into the interstitial fluid, there is another nice word to describe that. That's known as extravasation, because you are moving from the vascular system into the interstitial fluid. So you're going beyond the vascular system. Okay, so extravasation of leukocytes. Okay, so initially you're going to recruit neutrophils, which are going to phagocytose the pathogen. Later on, you're going to change. So after around 6 to 24 hours, what happens is the endothelial cells stop recruiting neutrophils to the site of infection and start recruiting monocytes instead. Whoops. Okay, so there is another type of cell within the bloodstream known as a monocyte. Okay, and this is the next target for the endothelial cells to recruit. Okay, so here is our monocyte. Okay, so this represents a monocyte. So basically, monocytes are circulating within the blood, and what will happen is after around 6 to 24 hours, these endothelial cells will start expressing molecules which will adhere to molecules on the surface of the monocyte. then what will happen is the monocyte 2 will diapodese across the endothelial cells and get into the interstitial fluid. Now, when it's in the interstitial fluid, what's going to happen is it's going to differentiate into a different cell type. Okay, so it's going to differentiate into a cell type known as a macrophage. Okay, now macrophages too are phagocytes, but they are they're a more expensive phagocyte than the neutrophils. So if neutrophils are the pawns of our chess game, macrophages maybe are a rook, so they're more powerful basically. Okay, so this process of the monocyte turning into a macrophage is differentiation. Oops, okay, so macrophage literally means big eater. That's what phage means, eating. Okay, and macro means big. So this is a cell which is going to come into the interstitial fluid and start phagocytosing these pathogens, basically. Okay, so that now concludes the acute inflammatory response. Okay, so we have recruited these leukocytes, neutrophils, and then later on macrophages, which are going to attack our pathogen. We've also brought in coagulation factors to try and contain the pathogen, and also complement proteins to directly attack the pathogen. Now what's going to happen is you're going to start the adaptive immune response. Okay, so if the innate immune response fails to clear the pathogen, then you're going to develop an adaptive immune response. Okay, and we're not going to go into this in any detail either, we're just going to summarize it. So basically what will happen is professional antigen presenting cells will take antigens of this pathogen to T cells which are in the lymph node. Okay, so professional antigen presenting cells, which I will just abbreviate as APC for antigen presenting cell. So this is some antigen presenting cell here. Okay, so APC stands for antigen presenting cell, and these are usually dendritic cells or macrophages. Okay, dendritic cells are the main antigen presenting cell which is going to initiate the adaptive immune response, and these are going to take antigen which they have found that is associated with this pathogen that we're trying to fight. They're going to take it to lymph nodes and present it to T cells. Now we need to understand that there are two types of adaptive immune response. There is what's known as the humoral adaptive immune response, and there is also the cell-mediated adaptive immune response. Okay, now the humoral immune response is to pathogens which are extracellular. Okay, so what do I mean by that? Well, you have to remember that we, as an organism, a human being, is actually made up of 100 trillion cells. Okay, so in actual fact, you are not just one life. You are in actual fact 100 trillion separate lives, okay, that are all cooperating to make what's known as a meta-organism. So human beings and other animals and other creatures which are made up of multiple cells, so plants and fungi as well, and metazoa, any sort of metazoa, okay? And metazoa just means that you've got multiple cells making you up. So where shall I write this? Okay, so metazoa, if you ever hear people use this word, it just refers to any organism which is made up of multiple cells, okay? And a protozoa, or a protozoan, is the singular. So metazoa is the plural, and metazoan is the singular. Protozoa is the plural, protozoan is the singular. So a protozoan is a single-celled organism, okay? So bacteria are protozoa. There are other examples as well. So for instance, many protists are also protozoa. Okay, so for instance, the malaria parasite is a protozoan. Okay, whereas multicellular organisms are known as metazoa. Right, so we are, what would be called a meta-organism, basically, because we're made of multiple cells. We're actually 100 trillion protozoa all working together, and it's often compared to a city, basically. The cells are like little people in the city, and they're all working together to make this larger thing. And some people actually want us to start thinking of cities as living structures in their own right, in this sort of spirit, but we won't go there. The point is that there are two different types of infection. Either you can have a pathogen which is just sort of in the city that shouldn't be, so it's someone who's come into the city and shouldn't be there, someone bad, okay, or something bad. Okay, so let's imagine a tiger was to come into London. Then that would not be allowed, basically. We don't want tigers roaming free in London. Okay, so that is analogous to the humoral attack, basically, when you've got pathogens that are within the tissue fluid, i.e. they're amongst the cells, but they're not actually inside the cells. Okay, whereas a cell-mediated attack is basically where the pathogen is actually within your cells. So let me draw this. So a humoral attack is when you've got a cell here, and then maybe the pathogen is here. Okay, so this in red is the pathogen. Okay, and this in white is a normal human cell. So humoral refers to extracellular pathogens. They're not at all going to try going within our own cells. They're just going to try and sit in the tissue fluid alongside them. Whereas intracellular requires a cell-mediated response, okay? So this is when you've got some pathogen which is actually going to go within your cells. Okay, so here's your cell, and here's your pathogen within your cell. Now, viruses would be the main example of a pathogen that goes inside our own cells. However, they're not the only example. For instance, again, using this example of the malaria parasite, that's an intracellular pathogen. Okay, we also have tuberculosis and Listeria monocytogenes, which are bacterial intracellular pathogens. Okay, so there are these two different types of infection, and they both require a different adaptive immune response. So we'll start off with the extracellular humoral response. So basically, what's going to happen is this antigen-presenting cell is going to go to the lymph node, and it's going to activate, and this is very, very watered down, it's going to activate CD4 positive naive T cells. Okay, naive T cells. Okay, so in the lymph node you have absolutely loads of CD4 positive naive T cells. Okay, and these are called CD4 positive naive T cells because on their surface they have a molecule known as CD4. Okay, right. So, the antigen-presenting cell is going to search and search and search for a CD4 positive naive T cell which is specifically targeted for a certain antigen, basically. So it's presenting, it's actually presenting a fragment of the antigen to this CD4 positive naive T cell, and it's trying to find a CD4 positive naive T cell that's specific against that antigen, okay, that antigen fragment. Okay, so when it finds one, and all the CD4 positive naive T cells have a different antigen fragment that they're against, what will happen is this cell will firstly differentiate into what's known as a T helper 0 cell, okay, and then it will divide many times, okay, so it's dividing and dividing and dividing. Okay, so here's another one, and another one. Okay, and then what will happen is these T helper 0 cells will differentiate into what are known as T helper 2 cells. Okay, so T helper 2, and I suppose I should write some of this down. These are known as T helper cells, and we have different types of T helper cells. So initially it was the T helper 0 cell, but then it's divided and made a whole population of T helper 0 cells, and those have started to differentiate into T helper 2 cells. Now, these T helper 2 cells are going to help activate B cells. Okay, so again, in the lymph node, you will have B cells here. So let's say this is a B cell, or a B lymphocyte. You also often hear them referred to as lymphocytes. Well, basically, what's going to happen is this B cell will be specific for a certain antigen. So if it's specific for the same antigen that the antigen-presenting cell has has selected this CD4 positive naive T cell for. So we're talking about a single antigen here. Of course, in reality, when you're actually doing this, you'll have many different antigens that you're capable of producing an adaptive immune response against, and you'll be producing it against all of them. However, we're keeping it simple, so we're just considering a single antigen that was on the surface of our pathogen, or maybe even the pathogen was actually secreting. Okay? So basically, when you've got a pathogen that has this antigen associated with it, and it's infecting you, then what will end up happening is this antigen's level will go up within the bloodstream, and this will happen because firstly, potentially the pathogen is secreting it, or maybe it's just releasing it into its surroundings just out of control. Because, you know, if you're coating yourself in a molecule, some of them are going to end up getting away from you. You're going to lose some of them, and they'll just go into the interstitial fluid. Alternatively, when the neutrophils phagocytose the pathogen, they will often just release a huge number of antigens afterwards, basically, which will go into the interstitial fluid and then into the lymph vessels, and they'll drain to the lymph nodes where the B cells are. Okay, so we'll continue this discussion in the next video.